In Chapter 15.4, we're going to take a little bit closer look at the process of phagocytosis. We'll talk a little bit about how phagocytes recognize foreign cells and don't attack us, and then talk about how they engulf and kill microbes, and then we will look at some ways that pathogens kind of go around this process to avoid it, and then once we talk about phagocytosis, that leads to the end of inflammation when all of the microbes have been phagocytosed. So we're primarily talking about neutrophils, macrophages, and dendritic cells here that are doing our main phagocytosis. So they are taking microbes in and digesting them in this process. There are specific particles or pieces of the bacteria that allow phagocytes, the cells that do phagocytosis, to differentiate between self cells and the bacterial cells. So here you can see a macrophage. It's reaching out with these little kind of spindly tendrils, and actually the best word to use, I guess, is tasting or feeling different cells. I say taste because it's actually looking at different molecules that are on the cell surface. And there are molecules on our cells that macrophages recognize, and they realize this is not something I should attack. So this red blood cell and this monocyte, it's not attacking them, but it is attacking the E. coli bacteria that are around it here because they don't have the correct self markers. There is an additional process that can go with phagocytosis. There are some bacteria that make a very slimy, slippery capsule layer. And when a macrophage or something goes to try and engulf them, it slides off because it's so slippery. This can lead to needing to use a different process, which we call opsonization. This utilizes antibodies, which we haven't talked about yet, but antibodies recognize specific pieces of microbes. And antibodies can coat microbes, and they can do this even for these capsule microbes. They'll recognize the outside of this slippery capsule, and they'll completely coat it. And that gives macrophages basically a little handle to grab onto to engulf the bacteria. So now it has something to hold on to pull it in. So this is one way that the two parts, the innate and the adaptive, the antibodies, work together to deal with infections. Once a phagocyte gets a bacteria inside of the cell, there are several ways that it can go about killing it. It can fuse with a lysosome and use what we call oxygen-independent mechanisms like lysozyme or those defensin proteins to destroy it. Or it can use oxygen-dependent mechanisms, oxygen radicals such as superoxide and hydrogen peroxide, all of these reactive oxygen species we've talked about. They are very good at breaking down bacteria in this process. So there are two main ways that it goes about destroying them. As we've seen before, some bacteria, some pathogens can survive this process of phagocytosis. Coxiella. It has special properties that allow it to live within the phagosome. Some bacteria like Shigella and Listeria escape from that phagosome and get into the cytoplasm of the cell. They're hidden and they can live very, very comfortable lives. Some, like Salmonella, prevent phagosome and lysosome fusion, so they stop this process midway through. There are even some bacteria like Shigella that can trigger what we call apoptosis, which is programmed cell death. So they will essentially tell this immune cell to destroy itself, and then they will escape from that immune cell that way. So just because we have a mechanism to deal with pathogens doesn't mean they haven't evolved ways to get around this, right? Once we're done phagocytosing, right, we've got all the bacteria, or majority of them. Neutrophils stay there for only a very short period of time. They basically live for about 7 days. As they break down, they become the white substance in pus. Once a neutrophil is done with its job of engulfing microbes and destroying them, it will undergo apoptosis, that programmed cell death. It's no longer needed. It probably has toxic chemicals and lots of damage to it. So it destroys itself and the pieces get recycled. To clean up these dead neutrophils, macrophages, the other cell, actually comes in and engulfs the neutrophil itself and degrades it safely so that those toxic components don't get released into the rest of the cell. All right, so phagocytosis, we have ways to recognize foreign particles in the body. We have two mechanisms of killing microbes with them. One of them involves reactive oxygen products. The other is lysosomes and other proteins that can break down bacteria. There are pathogens that have found ways to escape phagocytosis, and that allows them to live in the cells. And once we're done with phagocytosis and all the microbes are dealt with, then inflammation is allowed to subside and we deal with those neutrophils. The macrophages come in and clean them up. Okay, before we go on to the next section, let's go ahead and do a little review question. Put the following steps of phagocytosis of a bacterial cell in the correct order. So go ahead and look at these steps, pause the video, and then come up with your answer, and then we'll discuss it in a moment. All right. Okay, so the best way to go about this is finding what would be the first step. So we have the pseudopods extend. That's midway through, right? Lysosomes fuse and deposit enzymes. That's still like in the middle. This is early on. So if we look here, bacterium binds to the surface of the phagocytic cell. That would be the first step here, right? We got to bind to it and recognize it. Then the pseudopods will extend around it and engulf the bacteria. We have invagination of the phagocyte, which traps the organism inside of there. And then finally, the lysosome fuses and deposits the enzymes into the phagosome. So if 3 is the first one, we look at our options, and it looks like there's only one with 3, which is A. So that is our order of events. In Chapter 15.5, we're going to take a little sidestep and talk about some other important recognition bits. We're going to talk about interferon, and then we'll talk about these toll-like receptors. So interferons are another class of small cytokine molecule that are made by a lot of eukaryotic cells. We've actually talked about these previously when we talked about antiviral therapy. We can actually inject interferon in. And interferons are generally in response to intracellular infection, so microbes going inside of the cell. There are two types of interferon. Type 1 is generally responsive to viral infections. Remember, viruses need to use a cell to reproduce, so they'll go inside of the cell. Interferon basically is a warning signal that says, hey, I have a viral infection in this cell here. I need response here. Type 2 interferons help regulate other bits of the immune system, including antibody production. So they are part of the innate immune system because they don't recognize the specific virus, but they do recognize that there is a viral infection. So they don't know what species or type it is, but they will send out a signal to alert other parts of the immune system. So they're not specific, but they activate innate and adaptive other parts. So type 1 is generally activated by double-stranded RNA, which we tend to see in viruses, and it has an endonuclease that cuts those things. It can cut other bits that are required to translate viral RNA. Type 2 goes out and activates other white blood cells, macrophages, natural killer cells, and T cells. This is going to rely on and increase the number of what we call major histocompatibility complex antigens. So we'll talk about this later in chapter 16, but these help us recognize self versus non-self. All right, we're going to start with a case study, and then we'll talk about what's actually occurring in here. So we have Bobby. He's 11 years old, from Nebraska, and his entire life he's had recurring life-threatening infections, mostly caused by gram-positive Staphylococcus aureus and Streptococcus pneumoniae. Again, we see another recurring infection thing, so this is a common theme in this chapter. The infections include meningitis, osteomyelitis, that's bone infections, arthritis, and septicemia, and things from S. aureus. So we have lots of different infections in lots of parts of the body. They try to run tests to find a cause for this, and really can't figure out what's going on. So they look at the normal tests. T lymphocyte cells, those levels look normal. Antibody response looks normal. So they test the adaptive system. Usually there's some defect in there. But the adaptive is functioning. They also look at monocytes, macrophages in his blood. That number's normal too. So when they look at the level of cytokines, which induce inflammation, they find that they are very low. So it doesn't seem like inflammation is being activated when he has an infection. They do more tests down this line, and they find that there is a series of receptors that we call toll-like receptors, but Bobby is deficient for those. He has some sort of mutation or something that does not allow him to make those receptors that function properly. Again, with this, there is currently no cure for this option. So the only course of action to keep these infections at bay is to put him on a long-term course of antibiotics. They probably stagger the doses with different antibiotics or use a combination therapy. But eventually, unfortunately, this is probably going to lead to his bacteria specifically developing antibiotic resistance. And that could be bad for him because he has no immune system to call upon to deal with infections. So what are these toll-like receptors and their related group, the nod-like receptors? Toll-like receptors, they illustrate how science is very derivative in its naming. There was a receptor that was identified and somebody called it toll. I believe because when you get rid of it, organisms die. But there are a whole bunch of these, and so they called them toll-like receptors. And what will really warp your brain is then somebody will find something that looks like a toll-like receptor, and they'll call it a toll-like receptor, and so on down the line, right? We have stupid naming conventions in science. But the toll-like receptors can be found on host cells, and they enable the innate immune systems to respond. And what do they recognize? Well, they recognize those microbial-associated molecular patterns, the MAMPs. So things like peptidoglycan, flagellin, double or single-stranded RNAs, lipoteichoic acid, probably LPS as well. So things that bacteria and viruses generally have in them. Okay, so it's very generic to bacteria and viruses and pathogens. It's not recognizing a specific species, though. So once bound to a MAMP, so the toll-like receptor binds the MAMP thing, like peptidoglycan or something like that, it actually then signals into the cell to start making interferon and that pro-inflammatory cytokines that will speed up inflammation. Now, because in our case study, Bobby was missing these toll-like receptors, there's nothing to send the signal into the cell to start these processes happening. So inflammation is not getting started. These receptors are found in the membrane and look outwards. There's a related group called the nod-like receptors. They probably look like a receptor named Nod. Again, not very original. These proteins look inwards. They're important for intracellular sensing of molecular associated microbial patterns. So they're going to trigger different signaling pathways, but they will trigger the immune system as well. So if we look at them here, right, we have a bunch of toll-like receptors. They're recognizing different things like flagellin. That's a protein in flagella. And they're going to activate a pathway that's going to turn on genes to make inflammatory molecules. There's also the nod-like receptors. They're inside the cell. They're looking for bacteria or viruses that get inside the cell, and they will also activate this process, just looking at different parts of it. And so the cell is going to make cytokines, and inflammation is going to start, and hopefully it will deal with the infection that way. One lucky thing about Bobby is that he still has his adaptive immune system. His adaptive immune system will start to recognize the different bacteria that he has been infected with, and he should start to build some defenses to them. So it is probably not a death sentence for him, but he will probably continue to get sick at periods of time for the rest of his life. We talked about interferon, some molecules that modulate immune responses, and then the receptors that can sense these molecular patterns, the microbial-associated molecular patterns. Okay, that's it for 15.5 and 15.4. In the last video, we will finish up the chapter.